Good evening, everyone, and thank you for your attention. My name is Peter Shaw, and I'm planning to spend about the next hour telling you something about the Springtails or Collembola as part of the Tanyptera project. So, first things first, I aim to three targets in this podcast: to introduce what the Collembola are, to explain techniques for collecting and handling them, and to give you an introduction, but no more, to the main FSC Field Studies Council key to this group of animals. It's not going to be definitive. At the end of this period, you'll have learned a lot, but it's not the full key. We can always discuss later where we go from here. So, begin at the beginning. Springtails, Collembola, what are they? When I was a lad, as a student, a zoology student in the 1980s, we called them Aterygota, wingless insects, which is fair enough. They've got 6 legs like insects, they run around like insects, but they don't have wings. Nowadays, our view has slightly polished. Collembola are certainly close to insects, but they're probably as close to crustaceans. And it's quite possible that the last common ancestor of springtails and the true insects was a Devonian crustacean with more than 6 legs. We think that these are among the oldest and least changed of all the terrestrial arthropod groups. They mold continuously throughout life. And there's a few things which we're going to keep coming back to. I'll introduce them quickly, then come back in detail. So many, but by no means all, have an escape mechanism, quite a unique escape mechanism, involving a thing which is usually called the furca, or sometimes furculum or furcula, but furca is standard, which appears to be fused legs on abdomen 4, which is not like insects. And this furca latches into a hook on abdomen 3, stores energy, and the energy can be released very quickly to give the animal a high propulsion jump. Not all springtails have a furca, and is not a diagnostic feature of the group. The diagnostic feature of the group is a projection on abdomen 1 for the ventral tube, or colophore, which interestingly gave the group its name. Collembola is, I gather, sticky peg in ancient Greek. So here's a diagram I'm going to come back to, but this shows you a fairly typical large springtail, not as large on the screen than in life, clearly a termocerid, but I'd just like you to note that we have a very insect-like body with an insect-like head, an insect-like antennae, usually with 4 segments. Then we have a thorax, which bears the legs, and just 3 segments on thorax: prothorax, mesothorax, and metathorax. Then 6 segments on the abdomen. On abdomen 4, we have this jumping organ called the furca, which has a manubrium, a dens, and at the end, a muctho, or little tooth. Notice also VT is ventral tube. Not all springtails look like that. There's a group which I loosely call the Sminthurids, correctly the Symphyte pleona, which are severely fused and frankly look quite weird, but they do have a jumping organ, they do have a ventral tube. Usually these are quite small, less than 1 millimeter. Just to show you one of these Symphyte pleona close up, to show what it looks like front on, we can see the underside of this alachma. We can see the jumping organ, and between that, strange little lump, that's the ventral tube. And in fact, in these animals, the ventral tube can stick out quite a long way, absorb water off surfaces. These animals can drink without using their mouths. Right, so an obvious opening question is, how many species of Collembola are there? And that's actually one of those questions that's easy to pose and surprisingly hard to give a good answer to. Currently, if you add up the global lists, with about 6,500. I'm not sure. So if I've got a job at all as a recorder for the Springtails, my job should be to know how many we have in the UK. And yes, I've got a list. And my list has 414 species names on it. However, if you look at this list of species names, quite a lot are frankly fictional, dubious, unvalidated, one-off publications in dodgy old magazines of things we may well have been juveniles of common species. And of the ones I actually think I believe, we've now got 372 species. Is this correct? No, I'm absolutely sure that figure is not correct. For a start, we have quite a lot of new species arriving with horticultural imports, garden centres and so forth. There's at least 5 new species to science popping around our gardens. There's at least half a dozen old species are actually going to get split down the middle by DNA work into multiple species. And there's one family I'll show you, the Onychiuridae, where we've got the other malign combination, but the field was plagued in the past with dubious, fictitious, unlikely, implausible species. And if you use the old keys, sometimes one half an animal keys to one species, another half keys to a different species, so too many. But molecular work suggests that there's half a dozen or more cryptic species overlooked, sitting in plain sight. So really, with Onychiuridae, I'm not sure if we're coming or going, but somewhere around 400 species in the UK, which is not bad. Not in the same league as Hymenoptera or mites, for example. But you know, it's not a bad list. If you have a nature reserve and you want to add some more species to the list, get a Springtail survey. That will add 20 species to your list, almost guaranteed. Another little bear of mine is that there are no standards for how you're meant to pronounce things. And so some of the names I come up with may not be pronounced the same way as other people do. I was at a conference a while ago and heard a chap from the good old US of A talk about a fungal genus called Psilocybe. It took me 20 minutes to work out what he meant as Psilocybe, is what I call Psilocybe. So this genus is quite clear how you spell it, and I call it Dicotoma. Then I met someone else who called it a Dichotoma. And I put out a mass email to experts around the world asking how you meant to say this. And someone else came back, oh, you mean Dichotoma? Dichotoma. Dicertoma. In fact, the only thing that people agreed on was how important it is to be consistent in naming. Anyway, I'll stick with my system of names, and other people may pronounce things differently. Legend has it that at one stage you could tell which laboratory a mite expert came from by how they pronounced certain genera, with one lab using one pronunciation, another lab using another. People ask me what's special about Springtails, and I had to say my PhD was in them. Okay, what else is special? Well, their unique selling points. Probably the best claim to fame is how little they've changed. So one of the most famous fossil deposits is the Rhynie chert in the Devonian about 400 million years ago. which contains springtails, which frankly look pretty much like modern forms. At the time, our closest ancestor looked a bit like a newt, maybe a large newt with teeth. Springtails have hardly changed, at least in their body form. What else? When they jump, I think probably their most wonderful claim to fame is they're the fastest spinning animal on the planet, and they can jump and somersault 200 rotations per second. There's a YouTube video I recommend you watch in your spare time with some extremely high-speed filming of springtails. Alan Smith needed to film at 10,000 frames per second to get the details of their jump. What else? Springtails are almost everywhere on land anyway, and typically 5,000 per square meter, maybe more. Typically, no one notices them, but they're there, they're just rather small. You can get much higher densities. So here, this pink C here is a swarm of a springtail called Ceratophyzella bengtsonii on horse manure. You can see at the ports of an entire wall going black with a swarm of these things. Not commonly, I've not seen it. What else? Some make quite interesting chemicals. Some get all the stuff I've seen. The biggest in the world, I have seen it in East Germany, is a thing called Tetra odontophora islanensis, which is about 9 millimeters long. It looks a lot like a woodlouse. I thought it was a woodlouse at first, but it's not. It's a springtail. And they make some quite odd chemicals, including phenol and aminophenols for defense. So don't eat them. I'm sure you wouldn't anyway. So I want to show you pictures and tell you a little bit about some 5 basic forms of what springtails come looking like. And these are what you'll find if you go collecting. I slightly loosely call them some English names. Forgive me, taxonomic purists might go tut tut tut. So the biggest ones, the most obvious ones, the most colourful ones, the ones that turn up first in pitfall traps, for example, are the entomobryids. Rather large, large meaning about 2 millimeters. Not very large, but larger forms, almost always with a well-developed jumping organ. Not always, but very often they have a distinct colour pattern. And there's quite a few of these where you can confidently identify them just by colour alone. Not many springtails you can say that, but some of them you can do. As I come back to these, I'll emphasise how the thorax 1 is reduced. The legs appear to come out of nothing on thorax 1. Then we have the podurids. I tend to think of them as a bit like teddy bears. Matthew Shepard calls them the podgers. These are, again, typically surface dwelling, the Podurimorpha, medium to large. They often have a furca. It's so pathetically tiny, you wonder why they bother. So the furca is so piddly compared to their back legs. You think, why bother evolve such a tiny little thing? These are also common, widespread, and routinely encountered. There's one I'll show you on the film shortly called Neonura muscorum, which is in every woodland. Xenillas occur up every tree. There's a bright pink one called Bilobella, which has turned up in the northwest quite recently, turned up in Marple outside Manchester. And that's a podurid. This is the group which I'm open to being ticked off for. I tend to call them Smintheurids, and I probably shouldn't call them. I should call them Symphipleona. Symphilopleona, you can see why it's hard to spell. These are the highly fused springtails, medium-sized or sometimes frankly tiny, usually coloured, invariably with a well-developed jumping organ. I've think any of the Spinthurids lack a jumping organ, even the one that lives in caves. If these have an English name at all, they're sometimes called the lucerne fleas, which is not a very good name. They're not just about lucerne, they're not fleas, and technically that's the name for one species, Spinthurus viridis, which is a greeny yellow thing commonly found on vegetation with a very subdivided antenna 4. When you see a pretty springtail smiling at you, it's almost certainly this species called Dicertermina. It's distinctive, common, and the face looks like it's smiling. It's not, obviously. Then we have things which I loosely called onychiorids. To the students, I call them blind white sausages. Technically, the Euidaphic Hodioromorpha. Euidaphic means fully adapted to the soil. They're white, they're usually blind. The jumping organ is occasionally present and tiny, but usually absent altogether. Which is the hardest group to identify. Not only are they technically difficult to identify, you need to clear them in lactic acid or alkali to look at details of the skin. Secondly, as I said, I'm not at all sure I believe the actual species names in the books, because we're a combination of many unreliable old species and many DNA suggested cryptic new species. But we can get into genus easily enough. Beyond that, for example, my PhD work was on Onychiorus armatus, which is now Protophorura armata. I'm happy to say that everyone knows what it means. Or you can give it 10 different species names. Stick with Protophorura armata. There's some smaller ones of these called Mesophorura, which are really tiny and they're not at all easy. But you will find these particularly in acid leaf litters. Then we have the Nelids, Euidaphic Symphylopleona, and these are tiny. If you see the middle photograph, it has a big springtail. That's not the Nelid. The Nelid is a little white blob, smaller than its antennal segment 2, just under its nose. And that is the species with the truly silly, stupidly inappropriate name of megalothorax. You might think megalothorax is a big thorax. Megalothorax is so tiny that even with a microscope, you can overlook the wretched thing. And Nelides, if anything, even smaller. These are very common, especially in caves, but they are easy to overlook. And they usually turn up at the bottom of the dish. You finish sorting out the sample and look, what's that little white thing? Oh, there's 10 of them. It's megalothorax. Yes, keep an eye out for them. Right, springtails. Are they in any way, shape, or form? Perhaps, sadly, for my research career, no, they're not. There would be much more money in them if they actually did some proper damage. There are a few cases where springtails are reported of damaging young seedlings or leaves on young plants. Occasionally, stems have been girdled. This is distinctly uncommon and usually involves high densities of animals in plant pots. And rather than starve, they chew on the leaves. At worst, it's a bit of a nuisance. There is one I mentioned before, Spinthurus viridis, which can explode to pest proportions, and it can eat clover and alfalfa. So here we are, the lucerne flea. And I'm delighted to report that after half a century, we actually found some proper damage by the species in Wales in 2014. With a very minor paper out of it. You can see that these clover leaves have little holes in them. That was high densities of Spinthurus viridis. So yes, they can cause a little damage, but you've got to look long and hard. In terms of human pathogenicity, approximation, not at all. There is one story I met in Steve Hopkins' book of an entomologist who mishandled his pooter and pooted too hard and got some eggs of a springtail, Isotoma olivacea eggs, somehow in his nose. And remarkably, the eggs hatched in his nose and some isotomids actually emerged, made him quite unwell. That's really unusual. I've never heard that ever before or ever since. There's one case I've heard of a woman who turned out to have entomobides in her scalp from a plant pot next to the head of her bed. Yeah, right. Bizarrely, you may well meet some people who talk about springtails causing them parasitoses. I've actually had someone contact me saying, I've got a springtail parasitosis. Really? And I did offer to collect samples from their skin, and I was nearly allowed to. Unfortunately, these are almost certainly illusory parasitoses. They're imagined. When the university insurers heard something about collecting medical samples from a psychiatric patient, they said, no, you're not. Pity. I've no doubt at all I got sampled her skin and found no springtails. Swaddle, they're imagining it. One of the papers about springtail illness mentioned their pupae. They don't have pupae. If you really look hard, you can find people who claim there's a photograph of a springtail in a skin flake, and here it is. Here's the skin flake, and there's a blank skin flake, and it's a skin flake. Then they use Photoshop and painted a springtail, and you can see the top right-hand picture is a photoshopped springtail. Really, it's not a springtail originally, it's a skin flake. These things are neither evolved to feed on skin, nor can they attach to skin, nor can they hang on to skin. They're fundamentally not evolved with skin pathogens at all. And if anyone tells you otherwise, find a polite way of telling them that they are delusional. Then I found this web page. I'll just read it to you, just to warn you to watch out for. So, symptoms of a springtail infestation: fatigue, brain fog, memory loss, mood swings, depression, joint swelling, fibromyalgia, visual decline, sleep disorders, and so forth. You get the idea. Not to get into fun details, these are old standard, unmitigated, paranoia-inducing lies. I used this as example in my statistics modules to illustrate the idea of pareidolia, our tendency to see patterns when no patterns exist. So just go back one slide, people see a pattern in this skin flake, which, oh, it's a springtail. No, it's not. It's you seeing patterns that aren't there. So springtails will not hurt you. Good. Where do we go to find springtails? Well, actually, almost anywhere on land. For the start, soils. They're classically thought of as animals of soils. And in terms of species richness, most Collembola live in the soil, in or below the leaf litter layer. Typically, highest densities in the upper layer, declining rapidly in numbers as one goes deeper into the mineral soil. And the deeper you go in the soil, smaller and the whiter the animals come. You would expect that contrasting soil or litter types would have contrasting Collembola communities. And this is sort of true. If you find sharp contrast, like an industrial site, go to an ash dump, go to woodland, yes, you can see differences. Having said that, the differences between sites is not as clear and as simple as with the plants. You can go to chalk downland and know it's chalk downland by the plants. You can go to acid heathland, know it's acid heathland by the plants. Doing the same thing with springtails, not so easy. not entirely surprisingly for the radiation that came out of Crustacea. Some Collembola are coastal. None of them actually swim in seawater. Only a few can survive tidal immersion. Few can. By far the best known of these is nowadays called Annelida maritima. The old books called it Lepura maritima. I think this is ubiquitous. Wherever you look for it on rocky shores, you can find it. Typically, what you do is you go down to boulders about an hour after the tide has gone out, and they fall out of the boulders out of cracks to hide. At high tide, they stay in cracks, and their water-repellent hairs act to trap air. They can live underwater for hours at a time. Almost certainly all around the UK, and what you see in the distribution maps is maps of where people have looked for them. Be aware that just because it's small black springtail in a coastal site doesn't mean it has to be Annelida maritima. So here's a photograph which I initially thought, oh, this must be Annelida maritima, from Lee Johnson in the Shetland. In fact, what Lee said is, but they were jumping. Annelida cannot jump. Annelida means no tail, it has no furca. And these, in fact, were the Hypergastrura, which also swarm, also are podurids. And there we go. So the motto of the story is, check them under the microscope. Don't just believe it. It's probably Annelida by the sea, but it might not be. How about in fresh water? Yeah, there's a couple actually live in fresh water. A Greenia bidenticulata lives under stones, for example. But far more, quite a lot, live on fresh water. Famously, Podura aquatica, but my experience, that's quite a rare species. Also, many species of Sminthurides. And here you see Sminthurides aquaticus. And these have a remarkably shaped gutter-like mucro, which catches the water film. They can jump off the water film. These are technically called the Neustonic community, which is the community of animals on the surface film water. To give you some idea how big these animals are, in the bottom left of my slide, there's the eye of a frog. On the eye of a frog, there's a white dot. That's not a glint of sunlight. That white dot is a Sminthurides aquaticus that landed on the frog's eye. Rather bizarrely, you can also expect to find springtails up trees. Not many species, but very dependably. And if you study canopy fauna, you will undoubtedly encounter springtails. The one that you're almost certain to find is Entomoblia albocincta. This was one of the early things found in old birds' nests by early ecologists. So yes, way up a tree, bird's nest is abandoned, then Entomoblia albocincta moves in. Other species found up trees, including the Tomocerids, Lepidocertus, Orcosella, and Vertigopus. Just one or two more. And quite a lot of springtails occasionally turn up in canopy communities, and you think, doing there? You're going to be a soil species and you're 10 meters up in the air. So you can get these things, but typically where leaf litter has accumulated and formed what's called a suspended soil. Then there's a whole community which enjoy dancing on leaves of plants. So particularly, I've mentioned Spinthurus viridis, also Deuterosminthurus and Borletiellers. When Steve Hopkins wrote his book on springtails, he mentioned that as he worked in his garden on his computer, there's continual gentle stream of Deuterosminthurus palipes falling out of their bush onto his laptop, probably getting carried away in their mating dance. Again, this is on YouTube, it's worth looking up. Deuterosminthurus have a dance where the males and females put their heads together and literally dance on the leaf surface. in the end, the male persuades the female to sit on his spermatophore, and she absorbs his sperm that way. And yes, when you vacuum leaves, you'll find these species. You may well find new species to science. So the gardens in Richmond Park had a new species in the Catianidae. I do not know what to call this thing. It's one of the banes of my life. People keep finding these new species to science and say, Peter, what's this? I wish I knew. I really wish I knew. I contacted the word expert. She said, oh, I'll tell you what it's not. Thanks, Penny. Have a tell me what it is. Where else can you find springtails? Another place you might find them is this specialist community if you look in ant nests. So there's the idea of a myrmecophile is an animal which lives in ant nests. Normally, animals get torn to pieces if they're foolish enough to go in ant nests, but a few can survive. And if you study ant nests, I guarantee you'll find this white springtail, Cyphoderus albinus. Quite big as springtails go, blind. And early entomologists recognized it as a distinctive species, talked about its jerking actions, very commonly found in ants' nests. Things as well. So I helped Elsa Robinson look at ant colonies, ant nests in the Lake District. We found lots of Cyphoderus albinus, so quite a lot Lepidocertus cyaneus, which wasn't expected, but yeah, they were there. The other habitat where springtails are a notable feature of the biodiversity is in the fauna in caves. So we have the concept of a troglobite, and these are species which are only found in caves. There are a few, but they seem to get all over the place. So in the bottom of this slide, you can see a species called Oligophorura shirtii. And this is one of the blind white sausages, one of the onychiurids, which seems to pop up in caves and mines. So I've seen it in caves from Yorkshire, seen it in mines from Manchester, found it in caves in Devon. And you think, how the hell does it get there? Because it's blind, it lives only in caves. How did it get from a cave in Devon to a mine in Manchester? I think the explanation has to be there's a big hidden population living underground in water-filled cracks and crevices, and occasionally get washed out in bigger spaces where we can find them. There are others, the Schaeferias, Schaeferia, well, Lindberghii or Emucronata, typically found in caves, and that's really quite a variable thing. So I don't know if there's one species or 10 there. Each cave seems to have a slightly different population of Schaeferia, and arguably they're different species. Arguably, they are simply variations in one form. There's probably one of the rarest animals in the UK, if we've got it at all, is Disarlopolites patricii, which is meant to be found in one cave in Devon. And I went down that cave, and did I find it? No, I didn't. It might be extinct, maybe I wasn't looking properly. In deep caves in Wales, there's some really deep cave systems in Wales, and there's some white falsomias which just won't key. I'm almost certain they're new to science, a cave endemic. I put them down as one of our endemic fauna, but until we get DNA work on them, we can't rule out as just a surface falsomia with lost some eyes. So down caves, if you're interested in cave biodiversity, you need to look at springtails. I'm happy to help you. Oh yes, and another unique selling point, the deepest dwelling land animal is a springtail, Plutomurus, which is found nearly 2 kilometers deep in a cave in Georgia. And this is not often I get the creeps off scientific paper. But reading the paper describing the species, I got the creeps, because the methods section described how they got down there. And it wasn't a case of going down a cave. It was a case of going down a cave with a load of diving kit, then diving into an underwater lake in this cave to go down to another exit to the lake, which is then a vertical shaft 1 kilometer deep. They went down this vertical shaft 1 kilometer deep with some more diving kit, stayed down there for the night, and went into another underwater lake, went through that, and down another tunnel, and down this second to third tunnel, losing contact. They found this remarkably deep springtail, and if I were them, I'd be having nightmares about not getting out, because you've got 2 cave dives just to get you home again. Anyway, they did it, and they came back with, among other things, Plutomurus, also new annulida, Juicilla, and a species with a wonderful name, Schaeferia profundissima, the most deep Schaeferia. I don't expect anyone mad enough to do that. I'm certainly not doing anything of the sort. But we do have cave springtails, and they are an interesting area for study. So, how do we collect springtails? The standard technique involves putting a sample of some springtail material, such as leaf litter or soil, into a very simple device called a Tulgren funnel. I've got a small video here which hopefully will explain it in a moment. You can also do vacuum collection. So here I am doing vacuum collection from bark. This looks actually eccentric, but it works quite well actually, because the Dyson has no rotating blades. The animals get sucked into a vortex. And because they're jumpy, they jump into the vacuum stream, then get collected. So that, although mad looking, actually quite a good way of collecting surface-dwelling springtails. And I have once been congratulated for keeping the trees neat and tidy. Yeah. Sometimes you really want to collect springtails and there's no easy way to get them out. What you can do is give them an artificial habitat. People have used artificial birds' nests. I find scouring pads work surprisingly well, actually. Put a scouring pad in place, wait a while, come back in a few weeks, and animals move in. And you put the scouring pad into a Tulgren funnel when they come out. This works well in places like up trees or down caves, where there's not enough soil to be collected. So here's a little video about setting up a Tulgren funnel.
Hello, my name is Dr. Peter Shaw. I'm currently in my laboratory in the University of Southampton, and I'm going to show you some basic techniques for extracting and handling and looking at the springtails or collembola. So behind me is a device we're going to use to extract some springtails from a leaf litter sample, showing you the basic procedure. So this machine behind me is called a Tulgren funnel, and it's essentially very simple. You have a set of funnels containing the soil samples, and you put below the funnels a glass collecting container with some alcohol in, typically 70% IMS, and then gently heat up the sample with an old-fashioned warm desk lamp. I should show you this sample processing with some leaf litter I've just collected. So bear with me. Here we have part of the funnel. Here's the desk lamp, and below it we have a collecting tube with a label in. I cannot emphasize how important it is to keep labels in everything. In the funnel we have some mesh, and this is to stop the soil falling down the funnel into the collecting vessel. It needs to be very coarse mesh because animals need to go through it. This is 1mm mesh. I've made some holes in with a knife to make the holes a bit bigger. So I'm now going to add some leaf litter. So here we have the loaded funnel. You check underneath it's clear, then put the tube in place. Don't put the tube in place first, it'll get mucky. And now we put down a desk lamp with an old-fashioned hot bulb and leave this gently to dry out the sample for at least 2 days. In my PhD we allowed a week. That's possibly a little more than necessary. Certainly 24 hours will not be enough. Between, let's say, 2 and 5 days would normally do the job. So it's now a few days later and we've extracted the sample and we have a collection of small animals that have jumped out of the leaf litter into the collecting vessel. We then pour the contents of the collecting vessel into a glass container. Here's what I use. And if we look at the computer screen, we can see quite a variety of different animals there. So, as they say in Blue Peter, here's one I prepared earlier. This is a mixed soil animal extract from some leaf litter, and you'll see all sorts of things in there. There's the long, thin, multi-leg bodies of centipedes and of beetle larvae, and there are mites. There are wood lice, which are crustaceans. There are spiders, lots and lots of mites, but mixed in with them are some animals that look like this. They've got 6 legs, so they appear to be insects, long antennae like insects, but also a jumping organ, and that makes them springtails. And this is how you collect springtails. You extract them from leaf litter, then you need to sort them by hand, typically with a Pasteur pipette to suck them up. They fall into alcohol, you store them in alcohol, they preserve well in alcohol.
So let's revisit a tomocerid springtailed look in a bit more detail. I've shown this picture once. There we are. Head, antennae, prothorax is the juice, mesothorax, metathorax, and 6 segments.
So now I want to look at a couple of the springtails in the sample a bit closer up to show you some of the key details. So these relatively big yellow ones have all the features that I will mention. They have legs on thorax 1, 2 and 3, and notice that the legs coming off thorax 1 appear to come out of nothing. Thorax 1 is reduced. Between the back pair of legs you can just see the ventral tube sticking out. Then we have 6 abdominal segments and a jumping organ or a furca. This species has rather long delicate antennae which are snapped off in this specimen, but this specimen above has them retained, and you'll see antenna 3 is extremely long. This puts it in the family Tomoceridae. And this one on the bottom similarly, you can see this antenna is long and curled. 3 legs, ventral tube, jumping organ. This species used to be called Tomocerus flavescens, the yellow Tomocerus. Annoyingly, it's now been transferred to the genus Pogonognathelus, so it's much harder to spell. Pogonognathelus flavescens. And one more really common, widespread species I want to introduce, which you're almost certain to find almost anywhere you look, is this thing with a clear black belt on abdomen 3. Also look at the remarkable antennae, rather multicoloured, stripy, and if you count them, the antennae appear to have more segments than they're allowed to. Antennae have 4 segments in springtails, but this one seems to have 1, 2, 3, 4, 5, 6. And what happens is that the basal 2 segments in this species, in this genus in fact, are subdivided, just to confuse matters. and worse, subdivided only in the adults. So the FSC key takes out this genus on the antennae and misses out the juveniles, because juveniles don't have subdivided antennae. So this is Orchesella cincta. Cincta means belted. So I think that this is the black belt springtail because of the black belt on abdomen 3. Just to talk the segments again. Thorax 1 is reduced, 2, 3, abdomen 1, abdomen 2, abdomen 3 has a black marking on it. Abdomen 4 is also quite large, but in this case 3 and 4 are about the same size. That's important. Again, this one here, thorax 1, thorax 2, thorax 3, abdomen 1, abdomen 2, abdomen 3, abdomen 4, 5 and 6 just visible there. So this is Orchesella cincta, the black belt springtail. So another sort of springtail in this collection, I always discovered looking a bit like a teddy bear, maybe inaccurately. It's blue, it's about 2 millimeters long, quite hairy, very common in woodland leaf litter, and has no jumping organ at all, but it does have a ventral tube. You'll also notice that the first pair of legs comes out of a clear separate segment, unlike the tomocerids. This is a podurid springtail in the Poduromorpha, known as Neanura muscorum, commonly widely encountered across the UK in leaf litter in forests.
So I need to talk about segmentation patterns. It comes up in the key later on. It's a crucially important detail that's easily missed. So I've explained how there's 3 segments on the thorax, 6 on the abdomen, but a common variant that you need to watch out for is the thorax 1 appears to be missing, and it's just the pair of legs gives away. The pair of legs appear to come out of nothing. That's a very useful feature. It takes out the Entomoblydae and Isotomidae, and is mentioned in the first step of the Hopkin key, or very early on. I think the first step of the Hopkin key is the thorax 1 is reduced or not. So you need to watch out for that. As we go through identifying these animals, you end up unavoidably paying a lot of attention to segments and counting which segment is where, because sometimes the segmentation patterns are crucial and distinctive and easy to overlook. So a particular pattern I want to introduce and get people to remember is in the isotomids, when abdomen 4, 5, and 6 all get fused together into one bullet-shaped segment. When you see that, immediately, look at this 4, 5, and 6. I'm sure it's 4, 5, and 6. Then it's the genus Falsomia. These are small, either white or grey, and can be very abundant in leaf litter, also in plant pots. So whenever I get a report of infestation in plant pot, it's Falsomia candida, which is also found in caves, also used in lab work. Almost every bit of acid leaf litter anywhere in the UK will contain Falsomia podrioculata. It's got 2 eyes, 2 on each side, or podrioculata, 4 eyes. And if you look at the segmentation again, thorax 1 is reduced, T2 and T3 are there, abdomen 1, 2, and 3 are present, abdomen 4, 5, and 6 are fused together. So a little video just showing this.
And another widely encountered common genus, by contrast to Neanura, shows a lot of segmentation fusion. If you look at this little isotomid, we see not just this thorax 1 highly reduced, the pair of legs come out of nothing. There's 2, there's 3, abdomen 1, 2, 3, 4. Oh, abdomen 4 and 5 and 6 all fused together. This is the genus Falsomia and is distinguished by abdomen 4, 5 and 6 being fused. It does have a jumping organ, a clearly developed furca there.
You've got to watch these segments. I can't emphasize enough how important it is to watch the segments. So sometimes alongside Falsomia, you get this thing which is now called Pseudisotoma sensibilis, where thorax 1 is the juice, 2 and 3 are there, abdomen 1, 2, 4 are there, then 5 and 6 are fused. When you see 5 and 6 fused, it's Pseudisotoma sensibilis. But you can see how it'd be quite easy to overlook that boundary between 4 and 5 and say it's a Falsomia. You've got to check it. In fact, the jumping organ is different as well. When you get your eye in for them, it's distinctive, but you've got to watch the segmentation patterns. And there's one more really important question about segmentation, which the key asks about, which is easy to get wrong, and I've got it wrong, and it's easy to misunderstand it. And it's step 1, 2, 4, when the key asks about the ratio of abdomen 3 to abdomen 4, and it's easy to get this one wrong, because you have to measure the length of abdomen 3 and abdomen 4 in the midline. And if you look at this rather quaint old drawing here of Entomobria nivalis, you'll see that in the midline, abdomen 3 is quite tiny, and abdomen 4 is really quite large. The multiple given is 2.5 in the FSC key. It's more like 4 actually in this case. And you think that's easy. Yeah, right. Unfortunately, when you look at them side on, abdomen 3 and 4 about the same length. Nearly the same, not quite, but it's not 2 times. And it's so easy to look at the side and think, no, that's not. And then you go to the wrong part of the key and you waste an afternoon trying to key the wretched thing down. It won't work because all the time you're in the wrong part of the key. So when you get to key step 1, 2, 4, asking about abdomen 3 to abdomen 4 ratio, look at the animal in a dorsal view, otherwise you're likely to go wrong. I've been so badly caught out that way. If you've got a really big abdomen 4, it's an entomobriid, Entomobria or Lepidocertus. One of my ex-students, Erica McAllister, said, it's like wearing a saddle. That's a really useful image. If you're wearing a saddle, it's an entomobriid, big abdomen 4. And this corresponds to big muscles for powerful jumps. So a big abdomen 4 in this family means big jumps. All large active surface dwelling species with big jumps. Then just be aware that there are some where the segments are effectively invisible. And this is the case of the Symphyllopleona, which I tend to call the Sminthiorids. I shouldn't. And in some of them, the division between the abdomen and abdomen 4 and 5 and 6 can be seen. In others, it can't. So sometimes 5 and 6 form a little tail-like structure. But just beware, in the Symphyllopleona, you tend not to see segments. Okay, so I've mentioned the key. This is the key. This is the aid gap or FSC key to the Collembola of Britain and Ireland, written by the late much lamented Steve Hopkin, who, to be fair, rather despaired of identifying the group. And like I say, whenever you see a springtail that's smiling, it's a Disotoma, and there we are on the cover. There's one of the Disotomas smiling. His aim was to have a key based on simple, clear, dichotomous questions. And having battled with a lateral key in a book by Hubbard to Grasses and given up with a headache, I applaud the ambition of having simple, clear, dichotomous questions. the question's not so easy to answer. I have met taxonomic purists who are a bit sniffy about it because on occasions the key uses colour patterns. And within a UK context, I think that's fine. Maybe in a global context, if you want to get a key for families that cover the whole of the planet, then maybe colour patterns aren't so useful. In the UK, it's quite valid to ask if a Lepidocertus is white or dark. It's quite valid to ask about the pattern on Entomoblia abdomens. And sometimes in the key, members of the same genus key down in different places depending on whether they're dark coloured or pale. Purists like to have the genus first, then key to the genus separately. It works. This key isn't perfect, and no key ever can be. New species keep appearing, and then sure, there are new species appearing, and there are species quite possibly in your back garden which won't key. Yeah. But it works, most of them, most of the time. So what I want to do now is go through some of the questions the key will ask you and give you knowledge to help you deal with the questions when you meet them. And sometimes they need no explanation. So colourless or coloured Z's, for example. But there's some ideas that you'll keep on the meeting and you'll need to know what they are. If there is a good ecological reason to skip, that's fine, do it. So, for example, at couplet 151, you have the choice of deciding, does the mucro have a very hard to see spine at the base of the mucro, or was it collected from the seaside? In that case, if you came from the seaside, it's mesenterotoma. If it came from the seaside, it's Entomoblia, almost certainly. You can use common sense. Moving on, there are some questions which I will emphasize. Segmentation patterns come up, of which the most important I've already mentioned is the deduction of thorax 1. And if thorax 1 is reduced, if the front leg appears to come out of nothing, it's Entomobliidae or Isotomidae. Then there are the antennae. Now, here I'm going to say something that seems contradictory. The antennae are always four-segmented, but often don't look it. In other words, evolution has added subsegments. The default pattern is shown here by Philosomia and by an Entomoblia, which is 4 antennal segments. Antenna 1 is usually quite small. The rest is usually roughly equal length. Antenna 1, 2, 3, and 4. Easy. Not so easy is the genus Orchestella, where antenna 1 and antenna 2 are subdivided. So it seems to have 6 antennal segments, but annoyingly only in the adult. So if you look at my diagram above, antenna 2 is actually quite clearly one segment. It's just got a line in the middle. Unfortunately, only the adults have this, and the younger instars don't key out properly in the key. So just beware, if you have an Orchestella, it may not key out properly. And you can waste an afternoon sailing around the Entomoblias and thinking, go on here, there you are. If it's got a black belt on abdomen 3, it's probably Orchestella cincta. There's a good rule for you. Some of the podurids have antennae which, to be honest, you can overlook, like a short, squat pile of tires. And here's Lathria piga longicita, with lots of long hairs and tiny, what little antennae. The Homocerids are easily spotted, the first approximation, because antenna 3 is huge. If you see a big springtail with a really long antenna, it's a tomocerid for sure. They do tend to snap off. It is also a group where you can get the species purely by the way the antennae curl. So you can see on this rather wonderful photograph of Pogononothellus longicornis that the antenna curls down into a total beautiful spiral, a total full circle. That is distinctive of this one species, Pogononothellus longicornis. Annoyingly, all over Scotland, there's a very similar species, Pogononothellus flavescens, which is also yellow, also has antennae that curl, but not quite into full circle. They stop just below the full circle. So you see multiple spirals with longicornis. In Scotland, with a partial spiral, it may well be flavescens. But long antenna 3, that gives you the genus Tomocerid of Pogononothellus. Another really easy distinctive giveaway is when you see a Spinthurid, a Symphylopleonon, where antenna 4 is hardly there at all, reduced to a thing like a little thumb. As soon as you see that, it's a dicotomid, probably Dicotoma or Dicotomina. You then need to look at the pattern on abdomen 5. Also, there's one called Chenothrix arthur. Also notice that in life, these animals have waxy filaments projecting from their abdomen. You won't see it in death. They dissolve in ethanol. I've never seen them yet, except on photos. Then one of the more tricky set of questions concern antenna 4 of the Spinthurids in the strict sense. So many of the smallest Spinthurids have an antenna 4 which is subdivided, and you've got to count them. And sometimes it's not easy. For Spinthurus viridis, lucerne flea, there are 18 segments in abdomen in antenna 4. If you see a really weird antenna, a bit like a deformed stag's horn antler, what you've got is a male of the genus Spinthurides, and it came off the surface of water, probably a pond. These males have the weirdest antennae of any collembolan, like mini antlers. They've got a hook-like projection, and they appear to be evolved to hold on to female antennae while mating on the water's surface. And because of this, the keys out males separately to females, almost uniquely. You will, I'm afraid, need to worry about the eyes. How many eyes? The eyes on springtails, if present, are always simple oxellia. They do not have compound eyes, individual light-sensitive patches. Many have none. Some have 8. None have more than 8. 7 is not meant to occur. It does occasionally. 0, 1, 2, 3, and 4 all occur fairly commonly. Zanilla have 5 eyes, Tomostus have 6. If it's a Symphylopleonon, it's got 8 eyes, hard to see, unless it's in a cave and blind, or nearly blind, and it could be either Polites with just 1 + 1 eye. These are not easy to see, and the key is a little annoying in the way that it asks you to count. It has got more than 1 eye, it's got more than 2 eyes, more than 3 eyes, more than 4 eyes, and so forth. They can be really difficult to see. Sometimes you need high power, cold light illumination, and sometimes some of the big hairy ones, the eyes are so hidden and the warty excrescences has been really hard to find. All I can say is, yeah, keep trying, do your best. Maybe clear it now in lactic acid. Here's what eyes can look like. This is an eye patch showing 8 ocelli. You may well end up looking at the feet to identify your species, and you need to know some terms. The springtail foot has 2 bits, 2 terminal sclerites, one called the claw, one called the empodium. The claw is the bigger part of the foot. The empodium, or empodial development, sometimes empodium, is a smaller part of the foot. The claw is always present. The empodium is sometimes absent. Be careful. There are species where the empodium is present, but very small indeed, and easy to miskey. There's a genus called Shoatella, which, well, it's distinguished by having a claw but no empodium. And time and again, people have sent me Shoatellas, and you look hard, no, it does have an empodium. I'm sorry, it's something else. Shophelia usually. But the point is that it's easy to overlook a small empodium. Next to the foot, you sometimes have what are called tenant hairs. Tenant hairs with a knob on the end. The knob appears to be adhesive. It's found in species which are found trees. They climb things. If you see these on an isotomid, I guarantee it's purple. I guarantee it lives up trees. It's either pseudoisotoma or vertigopus. And you tell this by whether abdomen 5 and 6 are fused or separate. In both cases, they're tree climbers. They have these tenant hairs. The key refers to scales, and some of the surface-dwelling entomobliids have scales, particularly visible on thorax too. And in Victorian times, the scales of the podurids were, like old Victorian term, podurids don't have them now, entomobliids. In Victorian microscopic magazines, people sold podurid scales as microscope tests, because if you can see the patterns on the scale of springtail, you've got a well set up microscope. Almost every tree I've sampled, if you look hard, you'll find an entomobliid which has scales. It looks just like entomoblia, but entomoblia does not have scales, and Willosia does. And Willosia is distinguished by these leaf-shaped scales. There's a genus named for them. The genus is Lepidocertus. Lepidocertus, from lepido meaning scaly. And the entire genus is shiny silver by their thick coating of scales. I remember seeing these in my PhD, like little drops of mercury shining as they jumped around the bushes. Sadly, the species are currently separated by present absence of scales on legs and antennae, and they come off and they're hard to see. And my DNA work suggests they're not taxonomically very useful anyway. But for the time being, we go with the scale patterns. This can get quite difficult because sometimes you need to use high power to look at the exact patterns of the ketotaxy. Ketotaxy is the layout of the hairs on the body. And clearly these are not hairs like mammalian hairs, they're outgrowths of the cuticle. A normal seta is pointed, smooth-sided, and not particularly long. The key refers to macrosetae, which are the bigger ones, and often you'll see some really big ones, maybe twice as big as the segment, next to smaller ones, the sensili. Notice on this segment from Ceratophyzella denticulata, you've got a pattern of smaller setae between macrosetae. These can be hard to see. You'll need to clear the specimen in lactic acid or in alkali to have a chance. Occasionally, you find setae which are not simple but have side branchlets like a skinny feather. And these are called ciliated macrosetae, and they can be helpful. They're especially common in entomobliids. Now, I made a mistake earlier in my PhD in thinking the ciliated macrochaetae said entomobliids. That's not true. Here is an isotomid, or Isotomiella minor, common, blind, widespread in soils, which has long ciliated macrochaetae. Difficult to see, worth being aware of. Well worth being aware of. So there is a distinctive sort of hair called a trichobothrium. Single trichobothria, multiple plural trichobothria. I've seen an old book called them bothotricho, just to really confuse matters. Trichobothria are ciliated sensory hairs that stick out at 90 degrees from the body. Just to be confusing, scorpions also have trichobothria and they're different organs altogether. So trichobothria matters when you've got an isotomid in a wet habitat, because an isotomid in a wet habitat, especially with a dorsal midline stripe, is probably in the genus Isotomurus. One time it used to be one species, Isotomurus palustris, which is variable. Then we got Isotomurus maculatus, which is a different color pattern. Then the DNA work came up with another half dozen species. I tend to talk about Isotomurus lat. All the Isotomurus species have these things called trichobothria. We think they're for detecting air movements. And if you look carefully under high power microscope, you can see them sticking out the side, but they're not easy to see, especially if you use a vacuum collection and the vacuum strips them off. So sometimes you have to go by other features. One more really odd little thing you wouldn't know unless you were told. So when you're looking at the Springtails, the key refers to things called SAA. SAA are the subanal appendages, or sometimes the appendices anales, which are the oddly shaped hairs around the anogenital region of females. Yes, that means the males can be identified, the imagers can be identified down to species anyway. These are to do with mating, and these come in very distinctive shapes. Some look like feathers, some are club-shaped, some are curled. So those are the SAA, the subanal appendages. And when you need to, they can be quite distinctive. And the jumping organ itself, the furca. The furca is usually easy to see, and it has three parts: a manubrium, a dens, and a mucro. And usually it's easy, either it's there or it's not. There are the few cases which I dislike where actually it's there and it's so hard to see it's not visible. So Photophorura armata, Onychiurus armatus, has a pathetic little vestigial fold on abdomen 4, which is said to be the vestigial remains of furca. My pet dislike is the genus Freesia, which has a tiny little furca, but it is there. And you key these things out, you think there's no furca there, and it won't key out properly. You look really hard under high power. Oh, it is there, and that's why I wouldn't key out. I've wasted hours that way. For Freesia, the thing to look for are 3 anal spines. 3 anal spines, it's Freesia. So going back to the furca, the manubrium, the dens, and the mucro. You often end up looking at the hairs on the manubrium and be very careful which side is dorsal and which side is ventral. How do you decide which side is dorsal? It's easy. You imagine that the spring is pointing backwards, then the dorsal side is upwards. So my diagram on the left shows Perdura aquatica with its furca extended. Dorsal side is upwards, ventral side is downwards. Not always easy to see, but that's the logic behind it. The manubrium can be short, stout, and stubby, as in Balastura here. Manubrium becomes the dens, and here we have a typical isotomid dens. So notice that there's one manubrium giving rise to two dentes or two dens. This is why it's called a pitchfork. Manubrium dens. They can be, as here, crenulated, got a kind of roughened surface. Not always. Usually much longer than a manubrium. Sometimes the patterns of hairs on it matter quite a lot. So finally, at the distal end of the dens, we have the mucro. The mucro will have teeth. You need to count the teeth. Sometimes just one tooth, often two teeth, often three teeth, occasionally four, not very commonly. And sometimes the fourth one, where is a fourth one, is a tiny little thing on top of the mucro. So you see what appears to be three teeth. You look really hard, there's a fourth one on top. So needs careful checking. And then just to really make life difficult, one of my favourite little species, Tanyptera volgranii, has got a manubrium and a dens, but no mucro. I mentioned anal spines. If you see anal spines, they're useful. We think they're for wedging into tight spaces in the soil, not quite sure, but a lot of springtails have them. The commonest pattern is to have two. So the onychiaurids have two sharp anal spines. Hypergastrurids have two big sharp anal spines. If you see three spines, it's the genus Freesia. If you see four spines, it's the genus Tetraacanthella. The Latin scholars amongst you might realize Tetraacanthella means four little spines. If you find a ring of spines, it's Proctostephanus, and tell me, because I think it's extinct. Those are the easy features. If you try to key down the blind white sausages in the family Onychiauridae, you'll need to look at things called pseudoxyli. These are orifices from which they secrete noxious chemicals. And if you upset an onychiaurid, you'll sometimes see them make what looks like a yellow creamy liquid which bubbles out of their body. And this is shown to be repulsive to pseudoscorpions and spiders, as clearly defensive and presumably quite toxic. And the orifices from which this toxin oozes are called pseudoxyli, meaning false eyes. And they have been used taxonomically. To see them, you need to clear in lactic acid. And I have to say, the more I learn about them, the less I believe them. I have seen, my supervisor, Michael Usher, has seen, Steve Hopkins has seen animals where one half of the animal keys down to one species, and the pseudoxylia on the other side are asymmetrical, keys down to another species. So I reckon maybe half the names in the onychiaurid list are invalidated by this. Then we have the PAO, the post-antennal organ. If you look hard at the head of a cleared springtail, you'll often see a strange structure next to the base of the antennae. And it's not clear at first sight what's going on there. We think this is homologous with the second antennae of crustacea, the crustacean antennule. We think it's a sensory organ called the post-antennal organ, and they can be very useful. They can also be very hard to see. So when you do see them, they click into focus, and oh, they're very useful. So you can see this ring here in Annulidella, clearly visible. In the onychiurids, the inner structure of the post-antennal organ is really important. An entire genus, Tutiflora, is separated by having finger-like projections on the sense organs. These need clearing in lactic acid or alkali, then examining under ×400, usually with phase contrast. I think that's all I wanted to say about the taxonomic questions in the key. I could go through the key in more detail, but I think the best thing is to say, explore it, have a go, then drop me an email and I'll do what I can to help you. What I would like to emphasize is how important it is to handle the specimens correctly. And the reason for this is that IDs are not as straightforward or clear-cut as in many groups. So as a bird watcher, you're not asked to prove that you saw a blue tit. As a mammologist, you're not asked to prove you saw a badger. If you claim to have seen a bilobella or claim to have seen a Tetraacanthella, someone may well say, I don't believe you, that genus has been split. I need to go back and look at your specimen to see what it is nowadays. That's fine. And Steve Hopkin did a lot of that. And in order for that to be possible, you need to keep the specimens safe. To be safe, they need to be in, I recommend in alcohol, at least 70% IMS. I recommend the stronger the better. Steve Hopkin didn't like 100%, he suggested 95%. In my experience, 100% absorbs water quickly anyway. Anyway, what really matters is that its specimen is in alcohol, in glass, with a label that gives collection details plus a name. And this jar, this glass with alcohol in, should be in an outer jar also containing alcohol. So a good solution is to put small tubes in a Kilner jar, upside down, and the Kilner jar is full of alcohol. These last extremely well. I've seen specimens collected by Richard Bagnall in the 1940s, which were as good as things I've collected a week ago. I've seen specimens I collected myself 20 years ago, which look as good as things I collected a month ago. So remember the mantra: in alcohol, in glass, in alcohol, in glass, and make sure they're labeled. And that way, if they should be safe and someone can check them. Or you can make slides, and permanent slides are really useful. They work. They're so convenient. You put a mark on the slide, you can find the animal again immediately. An animal in a jar might take an afternoon to find. Slide made of euparal is safe, it's reliable, and it'll last. But then you find the key step asks about things like a molar plate or ketotaxi, and you need to clear the body. And there are media that allow you to do this. PVA, I use my PhD, or Hoyer's medium. And what these do is they mix a mounting agent with chloral hydrate, sometimes phenol as well. And these are magical. You put your difficult springtail in the mountant, and it clears in half an hour. Put a cover slip on, and you can see what you need to see, and it stays put, and you can key it down. Beautiful. The first time I met this, this is magical. This is so wonderful. There's a problem. I would say you don't have to clear that way. You can also clear in alkali, or in lactic acid or other media. Arnie Fehlberg has a clearing agent based on formic acid. That's fearsome. And he giggles and says, yes, it needs to be, yeah, put them in there, count to 10 and take it out or they're dissolved. And they work really well for stiff things like big black hypergastroids. They're good for thick-bodied mites. But dear, some of them are aggressive. Even KOH, potassium hydroxide, it works well, but you've really got to take the animals out as soon as they start to clear. They start to clear, you take them out. Then you wash them with distilled water. Then you dehydrate them in alcohol over the dehydration series. And if you're lucky, it'll work. If you're unlucky, what happens is that your specimen dissolves. If you put the specimen in Hoyer's medium or PVA, it'll disappear. I guarantee my PhD samples dissolved into mesocrystals. There's a lot of really important specimens in the British Museum and Natural History Museum which are worthless because they were put in a Hoyer's mountain and you just see a mass of crystals. The animals dissolved, it disappeared. Useless. So for long-term storage, the Natural History Museum will not accept Hoyer's medium as a mountain. They'll accept Euparal or Canada balsam, which are fine and they work, but you've got to clear the specimen first, then dehydrate it, then mount it. So actually you're getting one specimen onto slide. That's several days' work, spread over several days anyway. But if you don't, you might lose the entire specimen. So that'll do for the time being. I welcome email contact about Springtails. I give my personal email here. My university email works as well, but I check it less often. There is a Facebook group called UK Collembola, which I recommend you join if you're interested. I also list a couple of web pages which give species pictures and distribution maps. The Hampton one is my responsibility. I don't maintain it anymore, but I'm trying to keep it updated on my hard drive. The web page called collembola.org is operated by Frans Janssens, and he's a very amenable chap with amazingly good photos and very helpful. And I'll say, if in doubt, drop me an email. I'm delighted to help. If I'm in the country, I'll look at your specimen for you cheerfully. If I'm not in the country, you might have to wait a little bit, but I come back quite regularly. So I think that's all I wanted to say for the time being. I think that finishes my presentation. Oh, hey.
Great. Well, thank you very much indeed, Peter. Yeah, I found that fascinating, really. Listening to the ecology and your sort of funny anecdotes and stories around Springtails, and then it's going to be as a recording as well, really useful explanations, around using the key and things. That's really great stuff. Gold dust, really, for people interested in it and trying to get their head around going through the FSE key. So thank you so much. Well, now we normally do a question and answer session. So
I'll be delighted to take any questions anyone has, if anyone has any.
Great. So does anybody have any questions? I can't actually see any questions in the chat. So if anyone is sort of welcome to sort of put their hand up, virtually, and then sort of I'll invite them to unmute themselves and you can ask your question. But yeah, we don't have any questions at the moment.
Okay. Well, I'm always pleased to pass on. What? There we go. Good grief. What size mesh in the funnel?
Oh, and above that, actually, there's one from Alan Can. When are you going to update the key, Peter?
Yes. So let's take Alan Can's question first. I intend to add some couplets to cover new species and publish them at some stage, somewhere, somehow. But publishing the whole thing anew, I'm not sure FSC is up to that at the moment. Let's take the mesh question, that's much easier, which is that generally the bigger the holes, the better, because the bigger the holes, the more animals get through. But if the holes are too big, your tube fills up with soil. So it depends on your sample. If it's big leaves like oak leaves or sycamore leaves, you can get away with a centimetre mesh, it'll be fine. If it's a sandy soil, maybe 1 millimetre mesh, or you get a load of contamination in your sample. But generally, the bigger the holes, the better, compatible with not having a tube full of soil.
So do you take a range of meshes out then with you, Peter?
I suppose more by accident the design I have done, but usually it's been a case of buying mesh from Halfords and sticking my penknife into it to make the holes bigger.
Okay.
Simple old-fashioned techniques.
Okay, so there's a question there from Sean. I'll read it out. Is there any way to access the key without buying it? I normally struggle to get these specific books from the library annoyingly. Also, do you have any suggestions for identifying mites?
Okay, let's set the key question first. There is not an online version. I think FSC need the money enough. I'd say I would encourage you to buy it or get the library to buy it. Mite is another question altogether. I'd refer you to Matthew Shepherd. However, Matthew Shepherd looks at the Colin Bowler Facebook page and will take mite questions. So my answer for mites is ask Matthew Shepherd. He will send you a PDF. He will help you. I have despaired of mites. I'll tell you a little anecdote about mites to explain why I despaired. So I was on the guiding committee for the NERC thematic program on soil biodiversity, and I oversaw the Springtails. I also oversaw Lisa Cole's contract to do Springtails and mites. I thought, yeah, at last I get someone to help with mites. So I took a day off and spent the day with Lisa Cole doing mites. In one morning, we got one mite down to superfamily. Okay, people said maybe you could have done this, but you get the idea. After that, I'm not going to live long enough. There's just no way. So I gave up on mites after that. Maybe a kind of despair, and Matthew Shepherd has a good key. It is doable. With Felicity Crotty, they've got. I would encourage you to ask Matthew Shepherd this key. It's doable down to family anyway, but they're not easy. Springtails are much easier.
Okay, thanks. Thanks, Peter. Alan Can was just saying that a PDF of the updates would be great. Do you think if you publish some updates to the key, would you put them in somewhere that is accessible, or where would you normally go with something like that?
Publish it in a entomological journal and email copies to everyone I can think of, including your good self and Alan Can and Peter Boardman and anyone else who send me emails for Springtails. That's it.
Great.
I'm not quite there yet.
Okay, great. Thank you.
The key difficulty, literally a key difficulty in my key, is that the things I want to key out, my key, are in some cases new to science. We need to agree taxonomy of these new species before I can even get a key to them. So there's all over the UK, there's some spotty cattyanids jumping around people's gardens. And the world expert has said, oh no, it's not pseudocattiana. Thanks. Well, it's a start, but how about a name? So that's it. I'm not quite there yet. We need to have a taxonomy of these new forms. There's at least 6 new species hopping around.
Okay, great. Okay, so next question from Simon Dyer. Is the IDing process 2-part, i.e. try and identify under a microscope, then if unsuccessful, clear a mount on a slide and try that way?
It's not as simple as that. Some you can do quite quickly under low power. Some, I'm afraid, there's no choice but to either go to family or to say you got to clear it. And I just say follow the key through. I can't answer that question simply. It depends on the species.
Okay, so I guess, well, some you can just do straight away in the field, can't you? So I mean, just a
Generally,
spectrum, I
if
guess.
you're working in a canopy community, a suspended soil, the species you can do by eye. They're few, they're big, they're colourful. If it's a leaf litter sample, forget it. You need a high power microscope because there's lots of species, many of them are white. But there's a few habitats, suspended soil you can do, ant nest you can do, seashore you can do by eye. Soil, generally, you can't. That's the way it goes.
Okay, well, that's a sort of a good rule of thumb, when I think of it.
So thank you very much for helping me promulgate what little teaching I have on this subject, and it's been a pleasure and an honour. I just put a little chain of history, which is that I learned almost all of this from my PhD supervisor, Professor Michael Usher, who went on to Stirling University. He's now retired. He helped run the Bumblebee Conservation Trust for a while. And I just want to pass on his teaching to anyone else who listens, because that's the way academia works. You pass on ideas, and some get copied and some don't. I hope some of these get copied on. That's it. Thank you all.
Okay, brilliant stuff, Peter. Thank you so much, and hopefully we can get you up again at some point.
It's a nice idea, yes. I had a major medical issue, so it took me out of circulation a bit, but I'm mostly functional nowadays.
Great.
And
Yeah,
I like Liverpool. I like getting back to the Northwest. I'd love to go back to the Northwest and see my childhood haunts again. So I may well come up. I look forward to that.
Okay. Yeah, we'll speak more on that then. Great.
Okay. Look forward to it.
Okay, thank you, and thanks everybody for joining us tonight. Goodbye for now then.
Goodbye.
Bye everyone.